Assalamualaikum. I'm Asmad Kari from School of Animal Science. Today I'm going to give a brief introduction of Animal Reproduction Technology course. This is an elective course for Bachelor Degree of Animal Production and Health. This course is covering all current and anticipated uses of technology in animal reproduction to treat infertility and increase productivity of farm animals through enhanced control of reproductive function. There are nine main topics for this course, which are anatomy and physiology of male and female reproductive system, ovulation and estrus synchronization, cryopreservation of gametes and embryo, sperm and embryo sexing, artificial insemination, in vitro fertilization, intracytoplasmic sperm injection and embryo transfer, pregnancy diagnosis, cloning, and last one, student will also learn about ethical, moral, and social aspect of reproductive biotechnology. What is assisted reproductive technology? It is actually the reproductive technology used mainly to treat fertility in order to achieve pregnancy. In the first topic, a student will learn about anatomy and physiology of male and female reproductive system. At the end of this lecture, a student should be able to understand about the structure and also the function of reproductive organs in both male and female animals. Superovulation and estrous synchronization. Superovulation is actually the process of manipulating or stimulating ovaries by using specific hormones such as follicle stimulating hormone, prostaglandin, and also gonadotrophin releasing hormones to produce multiple oocytes in the ovary. Meanwhile, estrous synchronization is the process of manipulating the female's estrous cycle by using specific hormones so that they can be bred at about the same time. Cryopreservation of gametes and embryo. Cryopreservation is a process that preserves organelles, cells, tissues, or any other biological constructs by cooling the samples to very low temperatures to maintain their viability. As you can see in this figure, this is the simple illustration of method of cryopreservation using cryoprotectant or extender for male gamete, which is sperm, for female gametes, which is ova or eggs, and also for embryo. Sperm and embryo sexings. The genetic sex of an individual is set at fertilization, and it is depends on whether the egg-bearing ovum is fertilized by Y or egg-bearing sperm. The method for controlling the sex ratio is by separation of X-bearing and Y-bearing sperm. Many methods have been developed in attempts to achieve this goal, but in recent years, flow cytometry, as you can see here, has been developed to a point at which sperm sexing is available as commercial service. For example, in dairy industry, they prefer to have female offspring, so they will buy or use egg-bearing sperm for their AI program. Artificial insemination. It is the process of introducing semen into the female reproductive tract without contact of the female with the male by using specific tools for the purpose of achieving pregnancy. Artificial insemination is actually the most effective tools for genetic improvement as it could be able to maximize the use of superior males for reproduction and also for production purposes. As you can see in this figure, this is AI in cattle. The inseminator is deposited semen into the body of uterus by using AI gun. While in goat, the inseminator also deposited semen into the body of uterus by using AI gun and also with the help of speculum. Sometimes in small ruminant, laparoscopic AI is also being used for AI program. This topic consists of three different assisted reproductive technologies or ART as they are closely related to each other, which are in vitro fertilization, intracytoplasmic sperm injection, and embryo transfer. Among these three, IVF is one of the most widely known types of ART. It is a process of fertilization of the eggs and sperm outside of the body in a laboratory dish, as you can see in this figure. The second one, intracytoplasmic sperm injection, it is the process where a single sperm is injected directly into the center of an egg. For this technique, only a single sperm is needed for fertilization, and it is commonly used to treat infertility problems of males. The third one is embryo transfer. Transfer is the process of removal of an embryo from a female of superior genetics and the placement of the embryo into the reproductive tract of a female of average genetics. This figure shows simple illustration of embryo transfer process in cattle. Pregnancy diagnosis. Pregnancy diagnosis is an important tool to measure the success of reproductive management of animals. An early diagnosis of pregnancy is essential for reproductive management as well as economic production. In pregnancy diagnosis, the choice of method depends on the species, stage of gestation, cost accuracy, and also the speed of diagnosis. In these figures, um, we can see different types of pregnancy diagnosis in animal. The first one is palpation technique. The second one using ultrasound. And the third one, evaluation of pregnancy-associated glycoprotein. Cloning. Cloning is a process of making a copy of an individual. There are three types of cloning, which are DNA cloning, reproductive cloning, and also therapeutic cloning. DNA cloning is the process of copies of genes or segments of DNA, whereas reproductive cloning is a process of creating an animal that is genetically identical to a donor animal through somatic cell nuclear transfer, as you can see in this figure. Whereas therapeutic cloning is a process of creating a clone embryo for the purpose of producing embryonic stem cell with the same DNA as the donor cell. This stem cell can be used to create tissues that can repair or replace damaged tissues. The last topic is ethical, moral, and social aspects of reproductive biotechnology. The science of reproductive biotechnology has stimulated heated ethical debates among people all around the world, especially in regards to nature, religions, social, legal challenges, and also safety. So in this topic, students will be exposed and discuss about the impact of reproductive biotechnologies on nature, religion, social, legal challenges, and also safety. Okay, that's all from me. Thank you.